All right. Hi. Good afternoon, everybody. Welcome. I have about 200 students here. Well done to you guys. Hi, guys. Good afternoon. My name is Brian, and I'm representing IMU University today. We are bringing you live here on Zoom. And today we are going to be, running a series of SPM workshop, last minute preparation for you guys. And I think you're going to learn a lot from us here today. All right, you can see a lot of chatters in the chat. Hi guys, hi Chi Zhi, hi Alden, hi Sidel. Good afternoon to you guys. Once again, my name is Brian, and I'm representing IMU University, bringing you live here on the SPM final sprint. Okay, I understand and I know your first paper is next week. Yeah, even though it's the holiday right now, holiday season, how was your Christmas, guys? Today is Boxing Day. Hope you had received a lot of presents, but I believe you guys are studying hard, all of you. Yeah, studying very, very hard right now because SPM, one of the most important exams of your lifetime is happening next week. And, we are glad, IMU University is really glad that we are able to play a part in helping you guys out in preparing. IMU University, established in 1992, believe it or not, more than 30 years ago, is a premier private university on healthcare. So IMU University offers programs related to medicine, nursing, pharmacy, chiropractic, nutrition, dentistry, and so on. Yeah. So if you are interested, in becoming a healthcare professional one day, IMU University is definitely one of the universities you should be exploring. I'm so glad to be joining us here today. Because today's topic is on biology. We have Chigu Gan here. Chigu Gan is a very seasoned, very experienced teacher who have more than 30 years experience teaching biology in secondary school in Malaysia. And she's also an author for multiple books, including the Success Plus Biology and also other reference books. Yeah, so if you have a Success Plus book right now on biology, just flip onto the author page and you will see Puan Gan's name on it. Yeah. Thank you so much, everyone, for joining. I just want to set a few house rules with you guys. Kindly be kind on the chat box. This is a place for all of us to learn. If you have any question, please use the question box. Puan Gan will be addressing question on the final 15 minutes of the session only. Okay? Okay, we are almost approaching 300 students. So if you have, your classmate, your friends who are also taking biology, please let them know. Share with them the link. It's free anyways. Let them join in and let them listen into what Puan Gan have to share today. Okay, guys, I'm not going to take up so much time. It's already 2:04, and I'd like to pass the session over to Puan Gan.
Okay, hi everyone. My name is Puan Gan. I'm glad to be your teacher today. Is my voice okay? Brian, my voice?
Very clear, teacher.
Okay.
Very clear.
All right. So we will start off today. Let me just share screen. Okay, so I hope all of you can see the screen. Right, so before I start, I would like to indicate some of my preferences now. First of all, I would prefer all of you to write your question in the chat box.
All right, hi.
Okay, so I would also will address your question at the end of the session. So if you have any question, just write in the chat box. And I appreciate if you all don't disrupt by asking and interjecting question in between, yeah, so that I can have whatever that I want to put forward will be put forward in a very clear and smooth way, okay? So today, like I said, we only have 2 hours, or rather, I'm going to stop about 15 minutes before 4. So we have around 1.5 hours, and there's plenty to touch on. Now, what I'm going to do is not something that is for sure this is coming out. No, it's not like that, right? These are some focus point that you can focus on, and nobody know what's coming out, okay? We can only the best guess, that's all. And if it comes out, it comes out. If it doesn't, well, you still will have some knowledge of that topic or subject matter. Okay, so we shall begin, and it will not be, I will just touch and go for certain topics, all right? So we're going to look at certain selected topics, yeah? So we're going to start off with first chapter 1 and 2, chapter 2 for Form 4, chapter 1 for Form 5. Maybe you should pay attention a bit on certain basic thing like tissues, the different tissues that is found in our body, whether be it the human or be it in the plant. So for example, this is in the human tissue. So you should know the basic differences between the tissue and the function. Yeah. Say for example, L is skeletal tissue and it produces more energy. Okay, so I want to emphasize one thing here. If you see the word more here or not, see the word more or abundance. So your answer must have more. Now, mitochondria is a component cell that is found in every cell, but this tissue have more mitochondria because they need to produce more energy. So then you ask yourself, why do they need more? Because they need to produce more energy for the contraction and relaxation of the muscle. So the function comes here after that, right? So then you have similarity and differences between the two tissues. Like what is the difference between cardiac and smooth muscle? Okay, this is the answer here. Okay, so besides that, this chapter 2, now I have a lot of topics to cover. So I will just pick only those I think important. Now, the other thing you need to know is for Form 5, Chapter 1, is also a little bit about tissue. You have parenchyma, collenchyma, sclerenchyma tissue. You need to know the different type of shape and structure that this tissue have and their function. For example, parenchyma is a tissue that store starch. So they look a bit round and they have very thin cell walls. Unlike sclerenchyma, which has thicker cell wall that is thickened by lignin. So they have different function. One is to store starch. The other one is for mechanical support. So you need to know their tissue and their function. Okay, so I'm not going to go into Form 5, Chapter 1. So this is just a sample of question that is important, yeah? So I'm going to go straight now to Chapter 4, Form 4. Okay, so in this chapter, you know that it is about nucleic acid and all the chemical composition in the cell. So one of the important component will be nucleic acid, like how you must know how to draw the shape, especially the second polynucleotide chain should be facing downward, and where you should draw the line that link to the phosphate group and the nitrogenous base. So here you must draw exactly at the corners here, and the nucleotide here ought to be complementary, yeah? Ought to be complementary. That means if you draw like this, then the other one must be like this. So you have to complement it, okay? Okay, next is the phosphate group, and the rest you must know how to label them, okay? And then they will ask for comparison between DNA and RNA. So this, you need to know the similarities and the differences between them. Okay, so these are the differences of DNA and RNA, yeah? Okay, next stop is Chapter 5. Chapter 5, in Chapter 5, you know there is the process of condensation, hydrolysis. Before that, in Chapter 4, you know that you have this chapter 4 and chapter 5 is actually linked. So like, let's say maltose is actually the condensation between glucose plus glucose. So when you hydrolyze maltose, you get glucose plus glucose. So you should know all the formation, the condensation, and the hydrolysis process of disaccharide and also triglyceride. Okay, so I'm not going to go into that. And in chapter 5 also, you need to know how to draw the enzyme. You must know how to draw the complex enzyme molecule. Make sure when you draw it completely the same. The enzyme does not change shape, does not change size when you match it with a substrate, right? So it will form a complex enzyme substrate like this. Okay, so the size after that also, please make sure you maintain the size and don't change the size or the shape of the enzyme. Okay, so this one I will not go through because I think you already know the lock and key hypothesis of enzyme and all the factors that affect the activity of enzyme. Now, one of the important topic which I want to emphasize is immobilized enzyme. Now, immobilized enzyme is a new technique, is a new topic in the new syllabus that has not been tested yet. So I think it is important you all should know what is immobilized enzyme and its uses or application in industries. For example, in the production of lactose-free milk, milk contains lactose, which is then we use lactase, which is immobilized onto alginate bits. Okay, so these alginate bits are insoluble bits that will bind to the lactase. So when you pour in the milk that contains lactose, the lactase will hydrolyze lactose into glucose and galactose. And therefore, the milk that you produce is lactose-free. So the people who are lactose intolerant then can buy this milk and drink without any problem, without giving any problem to the stomach. Yeah. So the benefit of this is that the enzyme is not destroyed. It is more resistant to change in pH or temperature, and you do not have to incur more costs for separating the lactase from the product of glucose and galactose. Okay, so the enzyme will remain immobilized onto the alginate bits. Now the other thing is this: the use of pectinase and cellulase in the production of fruit juice like apple juice. So here we use pectinase and cellulase. Pectinase split or break up all the cell, okay, because the cell are bind together by pectin, which is a type of protein. So you add in pectinase to break the cell first, and then you add in cellulase to break the cell wall so that the juice can come out easily from the cytoplasm and vacuole, so you can get a clear apple juice, right? So they'll probably ask you other application of this technology in other food industry, like in the canning of fish. So you use protease. Protease is a general enzyme that you use for protein. So the protease will combine with the inert insoluble material, which is the alginate bits just now. So in this way, you make the enzyme more resistant to temperature change, and the molecule can also remain at the same position, and you can isolate or remove the fish skin much more easier. Okay, so these are the advantages. Okay, quickly now we look at chapter 6, form 4. Now, chapter 6, form 4, the basic mitosis, meiosis, you all ought to know already by now. The difference, the number of chromosomes, when they ask how many chromosomes in P or in Q, you should be able to tell the number, and also how they are arranged during metaphase or metaphase 1, okay, the usage of the word homologous chromosome and chromosome. Okay, so this one is very basic. You should know how to explain the differences. Next is, they might ask, like, what's the importance of mitosis in daily life, or maybe in industry or in agriculture? So you know some of the example are culture, tissue culture, meat culture. Okay, so for meiosis, the importance is to produce haploid cell, so that when the haploid cell combine or fertilize the gamete, they will form diploid zygote. Okay, so make sure you are able to draw. So drawing, okay, you can, if you have the free time, you go scout around all the topics that have drawing. Like, for example, enzyme is one of the topic that has drawing. This mitosis, meiosis also got drawing. Should practice a bit of drawing, because the trend now of late is drawing. So anything that you can see in your book, your textbook that has drawing, you should practice a bit, like phagocytosis, the formation of the food vacuole by amoeba, or the formation of after the auxin, you know, the auxin for chapter 5, chapter 6, is it chapter 5 from 5, you have the how auxin causes the coleoptile or the shoot tip to bend towards sunlight, towards light. You should know how to draw all the various type of treatment given to the coleoptile. So that kind of drawing you should emphasize on. And also like cell plate dividing the cytokinesis. So go through your book and look for any kind of drawing and practice drawing. Like DNA they might ask you how to draw a nucleotide, you know, so you all need to practice a bit the drawing. So like last year question, there were around, I think, 2 questions that emphasize on drawing. So that is a homework that you need to do yourself, yeah? Okay, next is a question on Down syndrome. Now, I think you all are so familiar with this question, but I'm just trying to emphasize what are the common mistakes that students do. First of all, you need to indicate what actually happened that causes this condition, which is the spindle fiber fail to function or fail to form, or the central fail to form. But most students put a full stop here without telling during when. So if you say homologous chromosome fail to separate, then you must say homologous chromosome fail to separate during anaphase 1. If you use the word sister chromatid, then you must say sister chromatid fail to separate during anaphase 2. So you need to indicate this. If you don't indicate when it happened, you will not get marked, even though you say homologous chromosome fail to separate. Okay, and then that will produce an abnormal number of chromosomes. Now, that's something that I need to tell you all. Now, make sure your answer is short, precise, and concise. Okay, so do not write a long sentence. Write short sentences, and try to keep 1 point, 1 sentence, 1 point only. Don't have the word and now. Don't use like, for example, the muscle of earthworm. Okay, if possible, if you want to say it contract, the circular muscle contract, and then the segment become thinner. Okay, so don't put and then when the longitudinal muscle, this is another point already. So put another sentence so that you don't have a situation where wrong cancel right. You know, your right statement earlier was cancelled away by your wrong answer in the second statement. So make sure you write short sentences, yeah? Okay, next. Are you all okay so far? Can you give me some thumbs up? Can you all understand? Are you all following? Okay, good. Okay, maybe I will pause after I finish from 4, yeah? Okay, next is chapter 7. Now, chapter 7 is one chapter that you really have to study, and it's not a very long chapter. It's a very short chapter. You have to study page to page, from page to page to the end of the page, because this chapter is very important this year. And you need to know the 3 different formula, okay? The 3 different formula you must know all the 3 different formula, which is aerobic respiration, lactic acid fermentation, and alcohol fermentation. Now, if you know this formula well, there's no way you can forget what are their differences, because the formula itself, from the formula itself, you can tell the differences, okay? And also the amount of energy, do not forget. Now, glycolysis is the first process here where it occurs in the cytoplasm, where the glucose breaks down into pyruvate. And then after that, the oxidation of pyruvate occur in the mitochondria to produce carbon dioxide, water, and 2898 kg of energy. Okay, so this is alcohol fermentation and then lactic acid fermentation. Now, do remember that a portion of the lactic acid is oxidized back to pyruvate by liver cell to complete cellular respiration in the mitochondria. The remaining lactic acid is converted into glycogen and stored in the muscle cell. Now, when all the lactic acid is removed, then only we say oxygen that is paid. Okay? When all the lactic acid is removed. Okay? Now, one of the things that students always make mistake is that when they say lactic acid fermentation, they are quick to say, oh, CRAM. Now, before you reach the final answer, most students do not start from beginning. That's why they miss a lot of answer. Okay. Now, if you, for example, this is one example, you explain the importance of fermentation. Okay, so they say produce energy, right, in oxygen limited condition. Okay, let's look at another question here. Okay, for example, like this, yeah. A man ran for 100 meter sprint, and then after that, the next thing is what happened to the man. And most students will say, oh, the man got cramp. The man got cramp. That's all. Full stop. Now, you need to say the high concentration of lactic acid that accumulate, high concentration of lactic acid that accumulate in the muscle cell, that causes the cramp, right? So this is what most students miss, high concentration of lactic acid. Or they will just say, oh, accumulate lactic acid. Okay. Next is differences between fermentation and aerobic. Okay, so this is the contoh above just now, where one is in the cytoplasm and the other one is fermentation. X here is lactic acid fermentation. So you have lactic acid and then aerobic, and then here is another example between human cell and paddy. Paddy are able to have high resistance to live in a waterlogged condition because they have alcohol dehydrogenase, yeah, that can break down alcohol into non-toxic compound like carbon dioxide. Okay, this is another example of differences question in this topic between aerobic and lactic acid. Okay. So you can see that they break up the answer, like occur in oxygen, produce water, produce carbon dioxide, and the amount of energy produced. So remember that aerobic occur in cytoplasm, mitochondria. This is only in cytoplasm. Okay. Next stop, we have chapter 9. Now, in chapter 9, like in the structure question like this, they'll probably ask you to name the enzyme secreted by one of the organ, and then what does it do, what's the function, what's the role of the enzyme, and the differences between the function of the organ found in the digestive system, right? Between R and T or S and T. Okay, so you need to know the medium, the pH medium, and also the substrate they hydrolyze, and also the product they produce in the different organ, yeah? Okay, next is this one. Okay, so we're going to look at one application question related to diseases or condition, yeah? So for example, this is a condition where the person have gallstone. Okay, the gallstone is formed inside the gallbladder and get stuck at the gallbladder, the duct, yeah? So they ask, the question asks, what's the effect of gallstone formation on lipid digestion? So the first answer is not to explain immediately, but to tell the effect on lipid digestion, okay? So that's why the first answer is digestion of lipid will stop. Do not attempt to explain first. Answer what happened here first. So after you have explained digestion of lipid stop, then you explain why it stopped. Why does it stop? Now, because the gallstone block the flow of bile into the duodenum, and therefore the emulsification of lipid cannot take place because there's no bile salt. So the lipid cannot be broken down into tiny droplets. So that's why the digestion of lipids stop, yeah? Okay, next, this is villus, the adaptation of villus for its function. So make sure you all mention fact plus explanation. Like for example, this one, numerous is to increase surface area for what? This is a function. This is structure or the fact, FACT, yeah? And then this is thin wall for what? Also for easier absorption. Network of blood capillary is always for transport. And then of course they have lacteal also for transport of fatty acid and glycerol, okay? Okay, this is one example of another question whereby they ask you to chart the process that involve lipid molecule from the time you partake the food until it get assimilated into the body cell. Now here is assimilation, because why? Because they say until it's used by body cell. That means you have to explain until body cell, okay? So only two things I want to emphasize here. Now you have, of course, first of all, talk about the process of digestion that takes place in the duodenum. And then after that, this is one point that students always forget, which is in the epithelial cell, the fatty acid and the glycerol condense again to form lipid droplets again. So which means that after digestion has happened, during the absorption into the villus, in the epithelial cell, the fatty acid and the glycerol will recombine again to form lipid droplets, okay? Then only it will be transported into the lacteal, and from the lacteal into the thoracic duct, and then into the subclavian vein, okay? So this is absorption. From here is absorption. The earlier part is digestion. And finally, the final part here is assimilation. So we can see questions sometimes ask these three components. In every food classes, okay, you have digestion, absorption, and assimilation. So your job now is to go and look out for protein. How does the digestion takes place? Where is it absorbed? And where does it go after that? What happened to amino acid? Okay? And glucose. So this is one example for lipid. Okay. Other question that can be related to digestive system are like, for example, the effect of taking too much of protein, excessive intake of protein over a long period of time, and what will happen to the cell, you know, long term will increase uric acid, form crystal in a soft tissue leading to gout, and what kind of food that should be avoided, okay? And then another one is this one. Sometimes they will ask what happens if the liver is defective or the pancreas is defective. Then liver is to produce bile salt that is involved in emulsification. So one of the first things that a person will face problem will be the digestion of lipid. But pancreas is even worse because pancreas is an exocrine gland as well as an endocrine gland. So if the pancreas is defective due to disease or something, now as an exocrine, it cannot secrete LAT anymore or less LAT, which is lipase, pancreatic amylase, and trypsin. And as an endocrine gland, it cannot secrete less insulin and glucagon. So that means you need to mention not just what it cannot do for digestion or what happened when there's 2 liters secretion or LAT and what happened when lesser insulin and glucagon is secreted. Okay. Now I pause here for a while. It has been half an hour now. Okay, let's see if you have any questions. I pause here for a while for okay. Anybody? You all can type quickly now. 1 minute for asking question. Okay.
There are questions, teacher.
Yeah. Quickly now. 2 minutes only.
Yeah. It's at the chat. Michelangelo Lai asks, teacher, may I ask when we are answering the structure and also essay question, can we answer it by using point form? Or are we allowed to do with 1 point with 1 line? Okay.
Essay, right? Let's say deforestation. It's easier if I give some example. You need to point form not mean you put it so brief until nobody know what you're talking about. Okay. You need to write the title first. Later, when I go into essay, right? Let's say they ask you what is the impact or what's the effect of deforestation. So you can write the title first. The effect of deforestation are ... and then you can write like that in point form in that way lah. If you mean that way, where you write sentence by sentence for each line can, but it has to be a complete sentence, not hanging sentence like example pollution. You cannot get marked by saying the effects of deforestation are pollution. What do you mean by pollution? At least you must say water pollution by what, what causes the water pollution. It cannot be hanging. You understand or not? It cannot be one sentence. Okay, for another example, the difference between nervous system and endocrine system. Nervous system is the transmission of nerve impulse is very fast, right? Whereas for endocrine system is slow. Okay, so this transmission of information, one is fast, one is slow. So you put in a table, and then you just put a heading. This is nervous system, this endocrine system, and then you put there fast, slow. This definitely got no mark. This is point form that has no meaning. What is fast? What is slow? Nobody know what you're talking about. So obviously this is out lah. So when I say point form, you have to have complete point form lah, like transmission of nerve impulse is fast, like that. That is okay. All right. Okay, I think I will move on from there. I hope Miss Michelangelo Lai, I hope I answer your question. Okay, so let's move on. The rest I will address it later. Okay, now let's look at one question here. This is about a nutritionist asking to explain the importance of planning a meal based on Malaysia Healthy Plate for individuals with health problems such as below. Now, if you look at this Pinggan Sehat Malaysia or Malaysian Healthy Plate, they are there for a purpose, not for you to just decorate the question. You have to plan meal based on this. First of all, you must know what is Malaysian Healthy Plate. Okay, it's quarter, half, quarter carb, quarter protein, and half vegetable and fruits. So when you say you plan a meal, you are talking about, like you go to a restaurant, right? You look at the menu and then you say, okay, I want my chicken roasted. I want my vegetable boiled. There is a way in which you prepare your meal. It's not just, or eat less carbohydrate or eat more protein. Not like that, okay? So you got to plan your meal. So specifically and targeted for the target group. For example, cardiovascular problem patient, you can't expect them to eat normal carb too much like white rice. So you got to suggest other form of meal that replaces the carbohydrate, the quarter carbohydrate. So here, example, you can say oat, cereal, brown rice, because why? So when you suggest it, you have to tell why. Like brown rice, why? Because it has low carb, low starch. And then vitamin and minerals, of course, from the fruits and vegetables, that one is without doubt. And then you have the protein. For example, why grill? Okay, grill because then grill has less oil. So you need to tell the method of cooking, like roast, boil, steam. These are method of cooking that reduces the amount of fats in the food. So, and then you suggest salmon. Okay, why? Because it has high omega-3. So if you can give the reason for the plan of the meal, that will be what is needed in the question, not a general answer as, oh, he has to eat less carbohydrate, eat more vegetable. Not like that. You won't get any marks at all, yeah? So please remember always, bio is not bahasa Melayu karangan. It is not just a general answer. You got to have facts when you answer. Okay, so for example, this one, brown rice, roasted chicken, cancer patient, maybe you want to suggest brown rice as well. White rice a bit is okay. Maybe they are recovering from a certain disease that they need a bit more energy than normal people. Okay, so that is how you address question that involve planning a meal according to measure healthy plate. Okay, next stop we have chapter 10. Now chapter 10, I'm just going to touch and go. Chapter 10, you have this one very important one, which is the mechanism of the contraction of the heart muscle. I'm sure you all know SA, no AV note, which I don't have to elaborate. And then the other one is another mechanism is blood clotting. Okay, so look out for these two mechanism, yeah? Blood clotting mechanism is another one, okay? Next, I have to move on because I got a lot more to share, yeah? Chapter 12. Now, we're going to look at chapter 12. This year, chapter 12, you see, when you look at the question, right, there are 8 structure questions, 2 essay, which you have to do. 8 structure question, question 9, question 10. These are essay that you have pilihan, and then you have question 11. Now, usually, like one of the essay will be form 4, one of the essay will be form 5, and wajib soalan can come from form 4 or form 5. Nobody know. Okay, so here, if you look at question 9, right, for human nervous system, it is quite important, either in structure or in essay this year for the form 4 section, yeah. So synapse is one of the questions that seems to be quite commonly asked in the recent trial paper in a lot of states. Many states ask about synapse, so you should pay attention a bit on synapse, how the transmission of nerve impulse is carried out through the synapse, yeah? And then, of course, the differences between sensory neuron and motor neuron. Okay, okay. Other important topics in this chapter would be voluntary and involuntary action. Okay, the differences between voluntary and involuntary action. Second one will be the reflex action. This is also quite commonly asked in the current trial paper, like the pulling of your hands from a sharp pin or from a hot object. And also the other one is the knee jerk. Okay, so at least know these few, they are very important, okay? All right, next, chapter 13. Now, I'm going to share now a question on graph, okay? So graph has not come out for quite some time in SPM. It used to be quite a usual trend that they will ask at least one graph question in the essay question. But of late, there don't seem to have graph, but in case graph come out, how do you tackle graph question? Now, if you ask me, sure, where got so many graph question in bio? Oh, there are graph question. In fact, the chapter 3 facilitated diffusion also can be converted into a graph form, and that's for chapter 3. And then you have this one. This is glucose. This is chapter 13, regulation question, glucose, insulin. Okay, and then you have which I will share shortly. Chapter 15, you have the staircase-like growth and the sigmoid curve. Okay, that's a graph question. Then you have your chapter 1 from 5, which is the annual plant, the biennial plant, and the perennial plant. That's a graph question. Okay, so there are some graph questions. And temperature body regulation can also be converted into a graph question. Okay, so how do you address graph question? I'm just going to briefly touch. Okay, if you look at this question, right, they ask you to explain the difference of the change in glucose concentration and insulin concentration between two individuals in 3 hours. Now, not only they want you to find the difference, they want you to explain the difference. Now, that is the tough part about this question. So when you look at this question, you'll find that this is the concentration of glucose. This is the concentration of insulin. Look at individual X compared to Y. Now, first of all, you see his glucose is much higher than Y. Even before he eats the chocolate cake, his glucose is already higher than the other individual. So individual Y, you can see that is normal range, yeah? So normal range is between 90 to 100 milligram per ml. So his blood glucose is normal. And then his insulin for Y, see this Y person here is blue color. His insulin seems to be able to increase in tandem with the increase in glucose. So it looks okay, you know, as the insulin, as the glucose increase, his insulin increase, but not for individual X. You see his insulin level is very low and his glucose level is very high. So from there, you can gauge really, oh, this person must be diabetes, okay? So from that, with that idea, we analyze the graph first, then you look for the peaks and the lows. So what do I mean by peak and the low? So if you look at here, during the first half an hour, he already increased. Okay, so this is the peak. And then how long does it take for the person Y to reduce? It takes half an hour, and then after that, the glucose decrease already. So within half an hour, his blood glucose decreased from the peak. So you look for the peak and where does it start to decrease. Okay, and where is the lowest point? So within 3 hours, he reaches back the initial concentration of glucose. So that's how you study. You divide the graph into different segments so that it's easier for you to answer. So if you look and compare with individual X, his glucose seems to increase even after 1 hour, still increase. After 2 hours, still increase. Doesn't seem to come down. After the second hour, only it comes down. So the time there is for you to draw your own imaginary lines there so that you can answer your question in a much more systematic way. Okay, so let's have a quick look. I'm not going to show you all the answer. Okay, so first of all, you see F1 here indicate the fact that you obtain from the graph. P1 is the explanation for the fact. Okay, so the first thing is his blood glucose is higher than that of Y. Okay, why is that higher? Because the question says difference. So you are comparing two persons now. Okay, so you cannot just state one person without comparing. Okay, so Okay, then the next thing is, after you obtain this from the graph, is to explain why is his blood glucose higher than individual Y? Because chocolate cake contains glucose. So the glucose spike up the blood sugar, okay? But then F2 is the second fact that you obtain from the graph. After that, after 0.5 hours, which is half an hour, the concentration of glucose continue to increase for 2 hours. Why? Why does it increase? Why does it increase? Then you explain because of the increased concentration of blood insulin and so on. Okay, so I cannot elaborate all. So after 3 hours, what happened? Because individual X secretes more amount. Okay, so Y has dropped back to original, but X is still higher because individual X secrete less amount of insulin. Okay, so that is how you answer a graph question. All right. Okay, I'm going to pause for a while. Any question? I'm going to look at the chat box for a while. Okay, anything else? If no, up to now, can you understand?
Teacher, there are questions in the Q&A box, quite a lot of them. 15 questions. Not sure if you want to address them now or later. Yeah.
Maybe I can address one or two now. Okay. Zingyi, no, in what way do you want to draw? What diagram are you referring to? Normally, diagram will not help in giving you more marks. Unless the question asks to draw diagram, then of course, but if it doesn't, there's no point for you to draw diagram in your answer. If you need to draw diagram, then draw it roughly in your rough paper. If that helps you to explain better, okay? Next, hydrogen bond, not important for you to draw. Just make sure you draw complementary. That means the two nitrogenous base must complement. Okay. Harriet is asking, sorry. Acid rain, I will do during chapter 10, yeah. How do we write amoeba? Amoeba is capital A, is exactly how you learn in chapter 8. Capital A, amoeba, and species, underline it separately, okay? And, okay, non-disjunction can be used at the same time. Why not just add the word, the one that I say homologous chromosome fail to separate during anaphase 1. It doesn't cost you anything if you were to write that, right? Okay, so sometimes they will give the mark for non-disjunction. Sometimes they will just give you mark for just explaining. Okay, for subjective question, if it's a table, like for example, let's say this is a subjective question, right? Now, you see, this is something not, how do I say? It's not something that is fixed. Okay, so I leave it to you because your teacher may say one thing, so I'm not going to confuse you. If you feel that you want to explain, like this is a different characteristic, right? You want to explain in words, let it be. If you want to put it in a table also, there's no harm. So there's no hard fast rule. Okay? So, but in essay, definitely in table, okay? All right? Okay, so now, question graph, my dear student, now I'm giving you the various topic. Whether it's coming out in structure and essay, no one knows. So you just need to know whether it come out essay and structure, does it make any difference if you have to answer them? So just answer lah, just know it. So I cannot tell whether a graph question will come out in which section, all right? So, but usually it comes out in essay, okay? Now I'm back to my question here, chapter 14. Chapter 14 here, label it properly. This is tendon, yeah? Tendon is attached to radius. This is ulna. Do not simply draw. Okay, this earlier I told you all need to know a bit of drawing, right? Remember I told you about amoeba and all that. This is another drawing you all need to know. How to draw the muscle when it's in this kind of position where the bicep is contracting. So you need to draw the muscle bicep a bit, fatter and shorter. Okay, but that's not the right term for when you answer. I'm just describing the picture here. And for tricep, you draw it longer. And the tendon, make sure you attach at the correct point. Don't simply attach. Muscle is attached to bone through the tendon. So this is radius. The bottom is ulna. So see the tricep here is pulled all the way up to ulna. Okay, draw it and pull it all the way to ulna, and here is to scapula and to the humerus. Okay. Okay, for drawing, and then after that, they will ask you the different characteristic between P and Q here. P and Q is tendon and Q is ligament. Can you see bone to bone? And then what happens if P is torn? Okay, so similarity, both are strong, but P, tendon is not elastic, while ligament is elastic. And then what happens if the tendon is torn? Now, before I answer that, let's look at how do you explain the bending of forearm? Okay, after you draw, right, they may ask you to explain the bending of forearm. Okay, so most students, they do mention P1. P1 is not a problem for most students, but P2 is a problem. A lot of students don't write that the pulling force produced by the contraction of the muscle actually is transmitted to the bone through tendon. This is one sentence that if you don't write, you miss out 1 mark, because usually the bending of forearm is 3 marks, usually. So this is one point that most students miss out, all right? Then the radius is pulled up, okay? Then if tendon is detached, or here they say tendon is torn, then of course whatever that I just explained cannot happen, all right? So it's a negative of the statement just now. Okay, next is the same, if whether it is at the leg or whether it is at the hand, okay, it's still the same, the answer is still the same. Okay, next is joint. Now, maybe they will ask you to circle, and what type of joint is this? This one, what type of joint is this? And then tell their differences, all right? So both are movable joint. And then, okay, this one allow, X is ball and socket, allow rotational movement, whereas this is movement in one plane. Now, in terms of their motion, this is circular motion, while the other one swing back and forth. Okay, related condition or diseases with this chapter is, for example, scoliosis and osteoporosis. What's the difference between a person who suffers osteoporosis and scoliosis? What causes it? Okay. And this is an example of osteoporosis question where they show you a photo of a person whose bone mass has decreased. So what is the food that you suggest suitable for this patient? So if you see that the bone mass has, a bit hollow here and there, is no longer compact, then you know the person is suffering from osteoporosis. Another related condition or disease is this one, which is osteoarthritis. Osteoarthritis, they ask the reason why the person has osteoarthritis is because of lack of synovial fluid, wear and tear, cartilage become thinner, okay, and the joint becomes swollen and painful, so the person have difficulty walking, okay? Okay, other question related to chapter 14 will be like, for example, this one, 2 differences between skeletal system in both these organisms here, grasshopper and earthworm. Okay, so one have exo, the other one endo. Exo is to protect body organ. Hydro is to maintain a rigid body shape. Okay. The name of the muscle. Now, at least you must know the name of the muscle in all the animal. You only learn about 4 animal, yeah. So it's not that difficult, yeah. Grasshopper is flexor and extensor. Earthworm is circular and longitudinal. And what are their movement? Okay, their movement is jumping. This is wave or peristalsis. Grasshopper use leg, earthworm use kete to anchor the body, okay? And then this is between human and fish, how their muscle contract, relax to produce movement. So for human is when bicep contract, tricep relax, but for fish is the myotum muscle. The left and the right myotum muscle will alternate to contract and to relax to whip the tail. Okay, that's all for chapter 14. Now moving on to chapter 15. Now chapter 15, right, is also pretty quite important this year. So if you ask me, based on trial paper, chapter 13 and 15, these two chapters are quite important. Now I missed out something in chapter 13 just now. Okay, I'm going to move back a bit to chapter 13. Earlier I talked about synapse, right? Synapse. Okay. Now that I remember, I better mention. Earlier I said about synapse, transmission of nerve impulse. Okay, and then voluntary and involuntary reflex action. There's another one for synapse. You need to go find out the effect of drugs, stimulant, and effects of depressant drug, and also effects of painkiller. What's the effect of these 3 different types of drugs on the transmission of nerve impulse across the synapse? Okay, and then chapter 14, beside this, chapter 13, beside glucose, another one I need you all to look at, which I don't have it in my slide today, is this. What is this? What is this? This is nephron. Okay, so please remember how the whole process of the formation of urine. Okay, formation of urine, ultrafiltration, reabsorption, and secretion. First, you must know how urine is formed. Secondly, is to go and do a comparative table between when you drink too much water, too much H2O, and 2 liter water. Okay, so remember the ADH until the urine form. Okay, so when you drink too much of water, then more urine, then less ADH. So go work out this table. This chapter is important, yeah. Chapter 13. Okay, the regulation of blood osmotic pressure. Okay, so I don't have time to go through this, so please do check it out. Okay, next is chapter 15. Now, chapter 15, like earlier I already said, comparison between a sigmoid curve and a life cycle of a grasshopper, okay, which is a staircase-like shape. The comparison between both, okay, in terms of their growth curve, and also for grasshopper, how does adisis take place? Like, for example, what is it that causes them to undergo adisis? Now, you already know that they need to go through adisis because they have a hard exoskeleton, right? They have a very hard exoskeleton, which prevent them from growing. So they need to undergo ecdysis to form new exoskeleton beneath the old one. So in order to do that, they need to suck in as much air as possible so that the old exoskeleton break and then they emerge with a new exoskeleton. So before the new exoskeleton harden, they will need to suck in one more round of air so that their length will increase before the new exoskeleton harden again. So the ecdysis, make sure you all know where it begin. So ecdysis begin here and ends here. So this is the instar stage or the NIM stage. Okay. So the comparison between both. Okay, so that is one graph question. Now, don't forget you have another graph question in chapter 15, which is the menstrual cycle. Now, the menstrual cycle story is so long, I don't think I want to take the time to explain menstrual cycle, but you need to know these three things that I'm going to say. First of all, you need to know all the hormones, FSH, LH, progesterone, and estrogen. So all these hormones, you can write short form, their function. What are their function? Okay. Number two, how do they affect one another? Like you have to use the word like if high FSH, then what happens? So you need to relate each hormone to the growing oocyte and also to the thickness of the endometrium wall. Okay. So you need to know what happened after ovulation. For example, this is the LH stimulate the remaining follicle graft to become corpus luteum, right? Then corpus luteum will secrete estrogen and progesterone that causes the endometrium wall to be thickened. So you need to know the function and their relationship between one another. And what happens if the woman is pregnant, then you draw that the endometrium wall continue to increase. And then this corpus luteum cannot degenerate. So you continue to grow, to secrete progesterone, estrogen, which also will increase. Okay. So, and then if this part here where menses occur, you use the word disintegrate, the endometrium wall disintegrate, but here corpus luteum is degenerate. And two things to help you all to remember what is the name for each of the graph is this. If estrogen, right, you will find two peaks before 14, after 14. That means before ovulation and after ovulation. We have one peak here and then another peak here when corpus luteum start to secrete estrogen. So if you see a graph with two peaks, then you know it's estrogen. Before day 14 and after day 14. Number two, for LH, you'll be increasing right after estrogen increase. So you'll be near to day 14. That's the second indicator for you to know that that is LH. Number three, FSH will increase below LH, not as high as LH, but slightly below. So it has two bumps like that. So that will help you to remember the hormone name. And lastly, progesterone is never seen to increase until the last part. After day 14, then it will start to increase. Okay, so you need to know what happened, how they interrelate, how they form the endometrium wall, and what happened if corpus luteum degenerate halfway during pregnancy, the effect. For example, if a person were to take contraceptive pill, what will happen to the wall of the endometrium wall? Okay, so that one all you all need to look up for it. Okay, before I go to Form 5, now I will again look at your checkbox if you have any, because I've already completed Form 4. Okay. Okay, any question quickly before I go to Form 5? Any question? No? Everybody can understand clearly what I'm trying to present now? It's been 1 hour, okay? All right, okay, so now if no question, we'll move on to Form 5, right? Okay, Chapter 2, Form 5. Now, earlier I've already mentioned Chapter 1, okay? So I'm not going to repeat myself. So Chapter 2, xylem, phloem, Chapter 2 plus 4 is important. 2 plus 4 because they are interrelated. There's xylem in Chapter 2, there's also xylem in Chapter 4. So both are interrelated, these two chapters. So first of all, know what is the structure and the function on xylem, how they are arranged end to end, okay, end to end, and how they are hollow in the middle so that they allow continuous flow of water from the roots to the leaf. The second one is the cell wall being thickened by lignin, okay? And then that provide mechanical support, okay? And then related to xylem and phloem is, of course, guttation and transpiracy or transpiration. So the differences between both, okay? Now, remember, earlier was all the Form 4 chapter. Definitely there'll be one differences. In question 9 is a Form 4 question, let's say. There'll be a differences question. In Form 5, question 10, let's say, there's also going to be a differences question, right? For example, difference between guttation and transpiration, okay? Now, in this chapter also, which you actually have done in paper 3, which is photosynthesis, but that photosynthesis did not tell the theory part of the production of oxygen. So you know, they may draw a chloroplast, and then after that, they ask you to explain the process in grana, in stroma, and the differences between light and light-dependent and light-independent reaction, okay? And then the difference in the internal structure of a leaf, and how it is adapted for photosynthesis. Okay, for example, the differences between palisade mesophyll and spongy mesophyll in terms of their arrangement, in terms of their shape. Okay, this is arrangement, this is shape. All right, and which one contain more chloroplasts? Okay, any question? This is xylem and phloem, and also translocation, which I did not write here. Translocation, don't forget. Okay. Now, I'm skipping quite a number of questions because I want to go towards chapter 10, 11, 12, 13, which are very important questions. So I want to spend more time in that, but that doesn't mean I never touch on 5, 6, 7, or teacher never touch, so, or not important. No, it's just that I have limited time and I want to emphasize some of the very important one, okay? So for example, chapter 9, Form 5. Chapter 9, Form 5, before chapter 9, okay, maybe I can indicate a bit. Chapter 8, what do you have in chapter 8 that is important? Nitrogen cycle. Okay, so if you look at the trial paper, there's so many questions about nitrogen cycle. Chapter 8, chapter 8. Nitrogen cycle and what happened when there are no decomposers. Almost, I would say, all the trial paper that touches on chapter 8 asks about the effect when there are no decomposers. Now, decomposers are saprophytic fungi and saprophytic bacteria. They are the one that break down complex organic matter into simple molecule. So they are the one that carry out ammonification, right? So they break down into ammonium compound. So if there is no saprophytic bacteria or fungi, then you know already the effect is that there will be less ammonium compound in the soil. That makes the soil less fertile. So the plant will also have less crop yield, okay, because there's not enough fertilizer in the soil. So, and then the whole nitrogen cycle, you need to know from the Rhizobium species to the Nitrosomonas, Nitrobacter, and so on, and the effect of the nitrogen cycle, okay? So that's all I want to mention about chapter 8. And maybe 1 or 2 questions about differences between archaea bacteria and eubacteria. So one of the easiest to remember is in terms of their cell wall. If it's archaea bacteria, A stands for archaea, eubacteria, archaea bacteria, and fungi, let's say, is their cell wall. Cell wall, this one, has no peptidoglycan, glycan. Eubacteria has peptidoglycan for their cell wall, and fungi is chitin. All right, so maybe just 1 or 2 questions asking about the characteristic of the different kingdom. Okay, you have the fungi, algae, archaea bacteria, eubacteria, plantae, and animalia. Okay, Okay, now chapter 9. Now, chapter 9, 9, 10 is overlapping. 9 and 10 is overlapping. Now, it can come out in question 11, right? Question 11. So, what are the kind of question that can come out for chapter 9 is about if the zonation of mangrove swamp, like for example, this one, you should know where is Avicennia sonarachia and not the rest. And then you should know their type of roots and their name. Okay, spell correctly, please. Make sure your spelling is correct. Spelling wrong, no marks. And the types of roots that they have. Okay, if you know the name, their name and their roots, that helps a lot in your explanation for succession. So colonization and succession in mangrove swamp. They may not ask the whole entire colonization and succession. They may just ask between how Bruguera or how Rhizophora grow in Avicennia and sonarachia habitat. Okay, so how did the zonation occur after, let's say, 30 years? Okay, let's say this is the zonation in current time, which is 2024. Okay, let's say in 2025, scientists project that it will move forward to the shoreline will move forward like this, let's say, and then Avicennia will move forward, all the plants will move forward, okay? So then they're asked to explain one of the succession process that occur between, let's say, Rhizophora and Bruguera, okay? So if you know the 4 names, if you know the 4 roots, you more or less going to get full marks already. All you have to do is to sandwich them between the names and the type of roots with the process of what the roots does, which is to trap silt mud causing sedimentation. So you know the root's name the root, and then tell the process of sedimentation and how it causes the soil to be firmer, higher, and drier. So this is the sandwich between the types of plant, right? So once the soil becomes higher and drier, it's no longer suitable for itself, but more suitable for the successor species. So that's how you explain. They won't ask you for the whole process. They'll probably ask for 2 types of plant only. So probably like 4 marks kind of thing. Okay? And then that is followed by this one, the importance of mangrove swamp, which is very, very important. So these are the importance of mangrove swamp. I already categorized them according to different area, which is the first one, protection area, improve clarity of water as a carbon sink, feeding ground, breeding site, forestry resources, and food and medicine. Okay, so for example, they may show an area that's covered by mangrove. And then the area was all cleared up, and then they start building some resort because it's near the seafront. So they built resort, and then they will ask you what is the impact of this project. Then whatever that is mentioned here will be the negative lah. What you have mentioned about the importance now will be the negative of what I've shown here, which is like, oh, they're no longer going to be a breeding ground for all the shrimp, crustacean, and all that. So it's the negative answer of what I'm showing here lah. So look at the question carefully. Okay, now this is another example of a question. This one, they said that the strong wave come and hit the housing area because there's no mangrove trees. So the solution is they build they plant mangrove trees and plant coral reef. So that is the solution that is suggested. So why do they suggest that? So the mangrove tree that is shown here is Rhizophora. It becomes a natural barrier that blocks the flow of water and lessen the impact of strong wave. So if you can have mangrove trees, it will help to break up the strong wave. So the wave that comes to the shore, to where the residents are, will be less strong. So it will not destroy the properties. And the coral reef that you plant underneath is to reduce the wave energy, okay? So these are some of the questions that they can ask you and ask you to suggest ways to overcome it. Now, chapter 10. Like I said, chapter 9 and 10 is overlapping. So we cannot tell what is actually coming out. It can be one whole question on chapter 9, or it can be just solely on chapter 10. Now, in chapter 10, right, there's this one question that is very important last year in last year trial, but it didn't come out last year for some strange reason. There were so many states that asked about food security in Malaysia, but it didn't come out last year. So I'm just putting it out in case it come out this year. One of the question in question 11, wajib, because this is wajib, that's why I need to emphasize a bit. If it comes out, if you're not prepared for it, you can't answer because it's wajib. Okay, so what are the components in food security in Malaysia? There are 4, AAUS, okay? Availability, accessibility, utilization, and stability. So for each of these, right, you need to know what it means. Like availability of food means what? Sufficient food supply. Accessibility means what? If food can be obtained easily by anyone, anywhere in Malaysia. Food utilization here means the ability of an individual, a housewife or a food handler in preparing food in the best way that ensure best nutrient for the people who eats it. And also food stability, regardless what happened, whether it's war, climate change, food is always there, available for everyone. So this is the food security, the 4 aspects of food security. Now, related to food security, they can ask, number 1, what's the impact of climate change on food security? Now, you know, climate change is a big thing now, carbon emission and all that. How would that impact food security? All right, so here you can insert in all the like pollution and all that. How does it affect food supply? Now, for example, if there's an outbreak of disease or there's a forest fire or something, now your food chain is going to get disrupted. This is one example. Food chain disrupted means the number of producers is going to decrease, right? So it's going to affect food production, okay? So, and if, let's say, if I want to make bread, I need flour, okay? So if let's say my wheat farm is engulfed by fire, I will have less flour, then I cannot make bread. So the food chain is disrupted. So food supply will not be there. So climate change is one huge thing that is affecting our food supply in the world, not just in Malaysia, yeah? Infertile land, continuous drought or continuous rainfall that destroys so much of agriculture. Even the recent flood also affect our destroy so many of our farmland and all that and our crops. So it has a great impact. So they may ask you, what's the impact of climate change on food security in Malaysia? Okay, that is one. And the second thing they will ask you is, so what's the government role? How to overcome this food insecurity? How to ensure food security is sufficient? So you go back to your AAUS, whatever you want to explain, right? You always refer back to the AAUS so that it will be a guideline for you to answer. You cannot, again, say something that is very general, you know, hoping that you will score mark. No, it doesn't. So, for example, availability. How does the government ensure there's available food for everyone? Affordability or accessibility. Now, example is the recent one, we have Menu Rahmah, right? Menu Rahmah may ensure that everyone has is access to food, cheap food that is affordable and is of high nutrient and quality. And then in terms of utilization, for example, then government efforts in educating the public on how to utilize the food that they have in access, convert it into something that is also, change it to another form of food that will reduce wastage and ensure like good practices in cooking, choosing the best food quality to prepare. Okay. And then, of course, other than that is food stability would be like helping farmers, giving subsidy, introduce latest modern technology in farming, intensive farming, so that we can get more crops in one season of planting, or maybe use drone to modern technology drone to apply chemicals, sorry, not chemical, fertilizer onto the plant. So these are some of the efforts the government can do to ensure food security. So that is one possible question. Another possible way is also using green technology. Now, green technology will be like for example, vertical planting. You utilize the wall that is unutilized, the empty wall, you can do vertical planting. You can encourage people to plant at home using hydroponic or aeroponic method. And then you can do one very important one, which is to plant in maybe greenhouses. Greenhouses would, you are able to control the factors that affect photosynthesis, so you can have more crop yield. Okay, you can control the temperature, you can control carbon dioxide, you can equip the whole greenhouse with irrigation, with proper supply of water and humidity. Okay, so that way you can increase crop yield. Okay, so this is one possible question on food security. Okay, next is, okay, I will stop for a while. Okay, no question, yeah? Okay, no question, then I will move on. Okay, next is this. Before I go to there's one question. Wait, let me see. I'll put it here. Okay, this one. It's in your textbook. I actually took this out from the textbook. This is one question that can come out, which is practices that contribute to environmental sustainability. Now, if you look at the current news, SDG is very, very important. This is another question, not food security. I'm done with food security. Now I'm talking about SDG. SDG, there are 17 SDG. You don't have to memorize them because they will probably give you in a stimuli form, and then they will say what are the ways to that goes help in to achieve that SDG goal. I hope you all know what is SDG, Sustainable Development Goal. Okay, so one of the questions that is related to SDG is this one. What are the practices? They ask you, what are your practices that can contribute to environmental sustainability? That means you want to make sure that whatever you are doing, using, will not cause further harm to the environment, but it will contribute to its sustainability. Okay, so there are few aspects here, so I will not elaborate. You can get this from your textbook, page 214. I'm just focusing on the topic only. Energy saving. Okay, that means converting the use of combustion of fossil fuel into something more like renewable energy. Okay, environmental friendly transport, 5R concept. That is the renewable energy. So the aspect is what I'm asking you all to remember. Renewable energy, environmental friendly transport, 5R concept, management of domestic and toxic waste, biological control method. And this is water saving. Here is harvesting rainwater, okay, to do all the household chores. So we like watering plants, so we don't have to waste water. Okay. Now, related to this, you can also add in green tech. Green technology is very huge, actually. The application of green technology is like, for example, you use a foliar fertilizer, organic fertilizer in place of chemical fertilizer, bioenzyme, Biomass is also another green tech. And then the other thing is biotech. Okay, biotech also can be applied. Like, for example, in chapter 4, you learn phytoremediation. So instead of using chemical to remove chemical in water, you use plant to treat waste water that is polluted with heavy metals like mercury or cadmium. So you use phytoremediation. This is in chapter 4, Form 5, phytoremediation. Why is phytoremediation useful? It's because you don't introduce chemical anymore to the water source. You will pollute further the water. So we introduce plant, and this plant, they are able to grow fast. They have long roots that can absorb all the heavy metal. So then after a while, you remove the plant, right? So like, for example, sunflower is used to absorb radioactive substances. So this is one method, one of the practices. Okay, remember, is what is it called? Practices that contribute towards environmental sustainability. So biotech technology like phytoremediation, and the other one is what? The other one is bioremediation. This is in chapter 13, Form 5. So in other words, right, in question 11 is no longer confined to one chapter per se. It can be like spread its question across a few chapters. Like, for example, this question is actually start off in chapter 10. You can end up in chapter 13. So you need to apply everything you know that you have studied that is related to environment, then you put it in. Okay, so it's no longer isolated chapters. You don't isolate the chapters. You have to overlap the chapters in order to answer question 11. Okay, so I'm going back a bit. Sorry, I've gone too far ahead. Now, other than that, right, you have a question like, okay, so first possible question 11 just now was food security. Okay, other than that, beside the textbook page that I show you, there are another method in which we can contribute to environmental sustainability, and that is the three things you all got to read up, which is preservation, conservation, and restoration. So preservation is what? To gazette, to protect, to preserve, and maintain. That is preservation. Okay, this is preservation. Conservation is what? Conservation got two, right? In situ and ex situ. So you have in situ means you leave the organism or all the components in its current habitat and protect it from being intruded and make sure that you will go back in number, or you remove them from their habitat, which is ex situ, and prepare an enclosure or conservatory center to house them, to make sure that you care for them, provide all the food and the necessity in order for them to recover if they're injured, or to allow them to reproduce in number before you reintroduce them back to their natural habitat. That is ex situ conservation. And the last one, restoration, is replanting or reforestation. Okay, so restoration here means you actively participate in restoring the condition that was once was. So, for example, if it has been a small area of hutan has been caught with fire, so you replant trees. You can also do that with mangrove. Mangrove, you can actively have gotong royong, go and plant mangrove trees. Or if you know scuba diving, then you go and plant coral reefs. Okay, so that is restoration. Now, this can also be applied as one of the amalan just now, the practices that contribute to environmental sustainability. Okay, so these are all the possible questions. Okay, wait. Okay, now we're going to look at third type of question, which is green building. Okay, how do you incorporate green technology in building a house or building, right? So you have to have elements in the green building that contribute to environmental sustainability. For example, a green building is a building that is use energy efficiently. So one of the things that you need to incorporate in when you build a house in line with green tech GBI, they call it Green Building Index, is to make sure that the house has as much as natural lighting as possible. So in that way, we reduce the consumption of electricity. So we allow natural lights. We have windows. We have a lot of windows. So we don't have to on light during the daytime. Okay. So that is one of the most important things, natural lighting. And windows that allow fresh air, good ventilation, so we don't need air conditioning. So it is a way of life in which we try to replace what we are so dependent on by way of building a building in a way that minimizes the usage of energy. Okay, that is the meaning to build a green building. Okay, and then you can also have roof that absorb heat, special tile roof that absorb heat, and then you can use also water harvesting system in the house. You have water harvesting system in the house, then you reduce the usage of clean water from pipe. And then you can also use solar panel on the roof so that you reduce the usage of electricity. Okay, another one you can do is, of course, have vertical garden, or even if you don't have a wall in the house, is to plant some indoor plants, or if you have a small garden, is to have outdoor landscape. So that contribute to better air quality in the house as well as in the environment. Okay, so if the house have better air quality, is more better ventilation, then you don't need to on so much of aircon, And then another one is you encourage housewife to actually make their own compost to kitchen waste, right? Turn it into compost, and then you use the compost to fertilize the vegetable or plant. So you reduce the usage of chemical substances. Okay, so that is some of the things that you can incorporate in building a green building, right? So that is the third kind of question. The fourth kind of question that can come out for question 11 is, like I earlier said, SDG. There are 17 SDG, so they'll probably pick one, like one of the most important one is clean water. If you go and look out for SDG, there's life on earth, overcome poverty, and so on. So one of the important one is clean water. Clean water by the year 2030. That was what in the United Nations Charter for SDG, the 17 SDG, one of them is clean water. So how do you have clean water? Ensure clean water in, let's say, in our local context in Malaysia. Okay, so these are the aspects that you can talk about. Education, educate people, and then usage of, ban the use of single-use plastic, like the red plastic bag is single-use plastic, use biodegradable plastic. So the less waste you produce, the less polluted the river is. And then you can enforce law, of course, and then improve our sewage system. Okay, so that means there will be less pollutant that enter the river. Ensure factories are not built along the river so that they will not let off their effluents into the river and pollute the river. Okay, and then remember just now I also mentioned about biotech, about phytoremediation and bioremediation. Okay, So that is for green, some of the possible question for question 11. Now moving on, I have very limited time now. Okay, is this one question in Form 5, which I assume all of you know how to do. This is probably in structure question. It's a genetic question. They give you a situation and then they ask you to solve. For example, in this question, based on this diagram, solve maybe what's their offspring, what's their ratio that their offspring will be hemophilia. Now, I just want to make sure that if the question did not mention draw a genetic diagram, just draw. Because when you draw, you get more marks than writing in words. So make sure you have your sidebar here. Everything must be written neatly. Use as much space as possible. Spread out your answer. Don't confine it, cram it. And if you don't write here and then you write here only, you get 0 marks. So make sure you write the sidebar here, write clearly all the aspect. Okay, so that will earn you at least 5 to 6 marks. Okay, now let's say the total mark is 8 marks. Let's say it's 8 marks. So you obtain 6 marks from the genetic diagram. Where is the other 2 marks? The other 2 marks is always to state first the disease that or whatever condition they mention in the question. Is it recessive or is it a dominant allele? And if it's a recessive allele, where is it found? Is it autosome or sex chromosome? That will give you 2 marks already. Right? Say for hemophilia is linked to the X chromosome. So mention it is a link to X chromosome. But thalassemia is not. So it's found on the autosome. So find out the dominant or recessive. And secondly, where is it found? Right? So that will be the question chapter 11. Moving on, chapter 12. Now, chapter 12 will be a question whereby you need to do a little bit of preparation. Okay, so earlier I said 3 possible, was it 3? About question 11, right? Now, another possible question that can come out for question 11 is chapter 13. Now, chapter 13 is the chapter whereby there are 4 things that you need to know. Number 1, you need to know insulin, how insulin is formed, and all other biotech technique like producing BT, how do you insert BT gene in a maize plant, for example. Okay, the method involved. Here, if you must look for DNA ligase, restriction enzyme, the method of inserting the enzyme into the plasmid, all that. The second one is DNA profiling technique. Okay, make sure you know the technique. The third one is, and what is it used for? DNA profiling technique is to identify a criminal and what else? Okay, the third one is bioremediation, which I mentioned earlier already. That is the use of bacteria. The fourth one is gene therapy. And the fifth one is advantages and disadvantages of genetic engineering. Okay, so related to this topic also, because these are in chapter 13 is the chapter 12. Chapter 12 is the genetic diseases, the different type of genetic diseases and their differences. Okay, like syndrome Down, this one is anemia with Klinefelter. What are their differences in their the disease? Yeah. Okay, this one is between Turner syndrome with Klinefelter. What else? This is I think this one is it's Down syndrome with Turner syndrome. So do a bit of homework. Okay, you cannot just sit down, just take a bio book and read like that. It doesn't work that way. You got to prepare some model answer and sit down and prepare the answer. Write it out by yourself with your own handwriting. You can remember better. And do a few differences like this between all these different diseases. What are their differences? You can find all this in trial paper, right? It's not exclusive. This one you can find all in your trial paper. And then what's the difference between monohybrid and dihybrid cross, autosome and sex chromosome? Okay, so the last part here is about, like I mentioned earlier, the chapter 13, gene therapy. Okay, the method involved in gene therapy. DNA profiling, the method. How do they carry out DNA profiling? Okay, how they extract, okay, the skin sample, whatever sample you obtain from the crime scene, you extract it, the DNA, then you cut it using restriction enzyme, you cut it into different sizes, then you place it onto the electrophoresis gel, which then will be separated, the DNA will be separated in by the electrophoresis gel, and then you transfer it to the nylon membrane, and finally you add in the ray film on top of it and process it so you can see the DNA profile. Okay, the DNA profile. So the DNA profile is unique for everyone. So if there's a dispute, for example, about the parentage of a child, let's say you must compare the band must match. This is the mother, this is the father, it must match the child. The DNA band must match. So therefore, this child belongs to this couple. If there's a dispute, for example, so you compare the DNA profile, okay? Okay, so, and then gene therapy just now is, gene therapy is for diseases like cystic fibrosis. Okay, so you normally will look for the normal gene first. Okay, this is just one name of a gene. So you find the normal gene, and this gene that you already isolated, okay, after you cut it with the restriction enzyme, you need to let it be transported by a vector. And the vector is usually a non-virulent virus. Okay, so this virus will then you inject it into the cell that has a abnormal gene. So the virus then will inject in the normal DNA into the cell, and then this normal DNA will start to produce a normal protein. Okay, so that will help to treat the disease. Okay, for example, if it's a cystic fibrosis, then the normal gene which is inserted will replace the abnormal gene. So then you can produce a normal protein. Okay, so the process is called gene therapy, right? So this is one process that you can utilize. All right, so I think that's all for my discussion today. So now I'm going to, yeah, it's 3:43 already, so I'm going to look at your chat box. So this is the time you ask any question, yeah? Okay, yeah, this is the time you can quickly now type in the chat box.
Yeah, teacher, hi. There are also some questions in the Q&A box.
Okay.
Yeah.
Can you list down all the chapters at the end of the meet? I think I have, right? From my discussion, I've already listed down the chapters. Well, anyway, I can quickly go through again. Chapter 2 is basic tissue, right? I started with basic tissue. Chapter 4 and 5 is always overlapping about enzyme, about DNA. Okay, and then chapter 6 probably about mitosis differences and the drawing. Chapter 7 is a must. You must read the entire thing. I told you the differences between lactic acid and aerobic fermentation. The chapters I don't touch doesn't mean you ignore totally. I'm just emphasizing some chapters, because some of the chapters, if you don't pay a little bit of attention, especially the question 11 one, I can guarantee you cannot answer. If you don't have a little bit more preparation for those topics, that's what I meant. So you need to prepare a little bit more, but does not mean you neglect the rest, yeah. Okay, chapter 9, I've already mentioned earlier, and then you have chapter 12, chapter 13, 15. You don't forget what teacher, why so many chapter 8? You got 11 question, you know. 11 question, and then 9, 10, 11 is sub type question, right? A, B, C maybe. So you will add in another 6, that will be 17 question really. Yeah, so then each of these question, like question 8, for example, it may overlap with another chapter. So you cannot like, oh, it's too much to study. Well, at this stage, right, you cannot say you have too much to study. You have come this far, you have to study, all right? So, and then from 5 will be 2, 4, and then you have your 8, 9, 10, 11, the later chapter lah. Okay. Okay, anything else? Anything else? Less insulin, all right? If they just say the pancreas is disease or part of it disease, you don't say no. No means the person also die already. Usually, if it's disease, then you say less, okay? Any more? I'm open to question. We have a few more minutes to go.
Yeah, teacher, there's a question. Teacher, can you see the Q&A box?
Yeah.
It's a Q&A.
Oh, Q&A.
You see it? A question mark. Q&A. There are 20 over questions there.
Oh, I was looking at the chat box actually.
Yeah.
Okay. Essay can be answered in table form, yes. What is OLAT is lipase, pancreatic amylase, and trypsin. Okay, share the slide later. Later, I will send to Brian.
Yes. Thank you, teacher.
I asked when we answer this structure. Okay, can we draw table now? I already answered that. You can or you cannot. Is open. It's an open question, that one. You can, and also there's no hard fast rule. Okay. Amoeba, underline the amoeba. Because the species is SP, you don't have the actual name of the SP one. You just write SP only, so you underline amoeba. Environmental sustainability and green tech. Green tech, actually, I've already covered so much just now. Draw table, subjective. Okay, again, it's a repeated question. The topic that come out during last year, is it less chance for them to come out again this year? Usually bio don't repeat. That's all I can say. Usually bio don't repeat. For the graph, I'll write the facts only. For graph, for the graph question, I will write the facts only and no explanation. Can I still get full mark? Of course, no. You write the fact without explaining. There's no mark lah. You can get the marks for stating the fact, but you cannot get marks for not explaining. PC, it is mandatory because it's easier for examiner to mark. If you want to help the examiner to not miss out any of your points, then do table. Because if you don't do table, you are going to make the life of an examiner very miserable because they have to look up for your answer, which is very tough. Looking into a computer screen, scrolling up and down to look for your answer. That makes them very, very, it is not advisable. Try to draw table. Please draw table. And encourage all of you to draw table for essay that compare and contrast. Make sure compare and contrast means similarities must be there. Differences also must be there. When you say similarity, don't put it in table, just put it outside like normal statement. The differences only you draw in table, okay? Okay. Differences between gene and chromosomal. Okay, maybe the last part later I will just quickly go through. Okay, English, linearly, English and BM. It's okay. Let's say you forget, right? Oh my gosh, I forget how to spell photosynthesis in English. It's okay. You can write in BM. We're not going to penalize you for writing just one BM word in the whole sentence of English. Okay, just write. It's okay. Okay. 360, 180-degree movement for hinge joint and socket. So far, I've not seen that kind of answer. Okay, so far it's always rotational and one plane. So use that. Anonymous, can you understand? Don't use unusual answer lah, which is never found, not the common answer. Okay, are there other answering technique when explaining why an offspring will inherit a certain disease? If a sex-linked disease and how... Yeah, I think I mentioned that earlier, that in order to obtain full marks, you need to mention that disease is caused by sex hormone, is linked to a sex chromosome or not, or is it found on the autosome? Is it a recessive or dominant allele? Okay. The dominant allele suppress and the dominant allele suppress the recessive. That is quite common term that you can explain. Okay, what else? Yes, wrong cancer right does exist. Wrong cancer right, yes, because for example, you state a statement that is contradictory in one sentence by putting an "and." You say two different contradicting answer in one statement. Of course, you cancel out the correct answer. Okay, so wrong can cancel right. Food web in structure question. Food web is so simple. You no need to focus. If you should, it's so basic. You learn food web in Sekolah Rendah. Okay. I should write meiosis or mitosis, or can I write specific meiosis 1? That depends on the question, whether they are asking meiosis 1 or mitosis. Dichotomous key is also Form 2, Form 2 syllabus, dichotomous key. So, Chu Ji Yao, refer to your science book, dichotomous key. Compare definitely need similarity and differences. Similarity do not write in the table. I already mentioned this the second time. Write it outside the table. Well, who told you that, Kam Zi Ching, that table has a high risk of getting mistake? I don't agree. Dialysis process is too long on Yan. I cannot agree. Function of a pedigree. It's not a weird question. It's to show the inheritance of the particular disease. That's all, Gary. Okay, so I'm going to go back a bit on, somebody asked about chromosomal and mutate. I got about, how many minutes do I have? Okay, so I'm going to quickly show now the difference between chromosomal and how you should study this topic. Okay, you have to draw a mind map, then it's easier for you. Okay, so the mutation part, right? Mutation, you have chromosomal mutation and gene mutation. Under gene mutation, you have 3: substitution, deletion. I'm going to use short form only, and insertion. Okay, substitution, you have sickle cell anemia. deletion, you have thalassemia. Okay, insertion is cystic fibrosis. So at least you must know a little bit about these two diseases. What are their characteristics? What are their characteristics? Thalassemia and sickle cell anemia. Okay, so gene mutation occur because of a mistake in one of the sequence of nucleotide, right? The bases change either in substitution, deletion, or insertion that result in a wrong amino acid pair. So when the amino acid is wrongly paired, then you get a non-functioning protein. Okay, so this is roughly the outline of gene mutation. Then in chromosomal mutation, you have two types of chromosomal mutation. One is increase or decrease in the number of chromosomes. And the other one is in the change in the structure of the chromosome. Okay, so under this one, right, you have two types. One is autosome, one is sex chromosome. Under autosome, you have Down syndrome, which is an increase in the number of chromosomes. Under sex chromosome, you have a few. You have Turner syndrome, Klinefelter syndrome, you have Jacob syndrome, and trisomy X, 3 X's. So these are the increase or decrease in the number of chromosomes. Now, this is caused by non-disjunction. I'm sure you already know. Okay, now, this one, there's only one example given in your syllabus, which is Cry-Duchet. Okay, Cry-Duchet is deletion. There are 4 types of chromosomal mutation due to the change in the structure of chromosome. Okay, so you go and look out for the 4 types, but the one that they emphasize on is only on deletion, which is Cry-Duchet. Cry-Duchet is negative 5p. The 5p arm is missing. 5 is chromosome number 5. P is the short arm. So the short arm of the chromosome number 5 is deleted. So since it's deleted, so the child born has some defective in their physical as well in their mental ability. Okay, so this is the outline of the diseases. So from these diseases, right, they can ask any kind of differences. They can ask what's the difference between SCA, sickle cell anemia, and thalassemia. They can ask what's the difference between Turner syndrome with Klinefelter. They can ask what's the difference between Down syndrome and Jacob syndrome. That's the homework you all have to do a bit lah. Okay. So these are the example of the question that they can ask in chapter 12. Okay. So if there's no more question, I think that's all. Wait, let me have a last look at the Q&A. LH, all hormone can be short form, but capital. No, you can draw figure. You yourself say drawing figure, so you can use freehand. Anonymous, it depends on the question. I cannot specifically say how you should write. Should you write mitosis, meiosis, or meiosis 1? It all depends on the question and the diagram they give. Oh, okay, Ching Wei, you asked a very good question. That is the common mistake that most students write. Eat. Actually, the eat, right, in the second sentence, right, sometimes we don't know what you're referring to. So it's best to repeat whatever eat you're talking about. Okay? You understand, Ching Wei? Repeat it. It's not going to be very long, right? The words that you are using. How can we answer KBAT question out of textbook? KBAT question it can be answered. There's no way they're going to ask out of textbook. It's going to be related. It's going to be just like, for example, one KBAT question that came out last year is, why do you apply ice on the swollen ankle? A boy sprained his ankle. Why do you apply ice? Now, the concept of applying ice is not something new for students. It's something that you all do all the time when you all practice in PJK. You all fell down. The first thing your teacher will say, go take some ice from canteen. So it's not so difficult. It's not about out of this world question. It's answerable. Those are keyword questions. Okay. How do you draw a chromosome? Chromosome, if it's at prophase, it's like that lah. This is one chromosome but consists of two sister chromatids. Okay, what else? XYY, the Y is during sister chromatid fail to separate. Yes, albino is gene mutation. I think I forgot to add albino in. Albino is gene mutation. Yes. Yes, WCR exists in assay. What do you mean? Do we need to write full name before writing in short term? I don't understand that. Sorry. How do you know the name of chromosome in different stages of cell division? During the first phase before prophase, they are known as chromatin. Then during late prophase, where you can see them condenses, then we call it chromosome. During my metaphase, anaphase, they are still chromosome, but we can also say the chromosome consists of two sister chromatids. That's all. Then during anaphase, when they separate, we say the sister chromatids separate. Then when they reach telophase, we call them chromosome. Okay? Yes, FSH, LH are all usable. Okay, I think that's all. Brian?
Thank you so much, Puan Gan.
Okay.
Very informative session.
I hope
You'll be sharing with me your PDF file.
Yeah, I hope they understand and benefit from today's sharing.
Yes. All right, guys. That's it. Guys, please, if you appreciate today's session like this, please type thank you in the chat. Give us a thumbs up. I just want to remind everyone. I just want to quickly go to a slide that I like to share, which is we are having another session tonight on chemistry. And guys, if you are taking chemistry, you do not want to miss this. Okay. All right, guys. Thank you so much. Thank you for being very cooperative in the session, being very respectful to each other. Thank you so much. And go for it. Hope you all score in your biology. Study hard. Take care, guys. Bye-bye.
Bye-bye, everyone.
All right. Thank you so much, teacher. I still see there are 40 students here. I'm going to close this room. So see you all tonight.
What's the attendance?
480 registered. I see about 400 at the highest point.
Oh, okay.
Yes.
All right. Thank you, Brian. Thank you for the opportunity. Bye-bye.
Thank you so much once again. Another very, very